materials have to be redesigned?
Hello and good evening to everyone. Thank you for joining us today. The Institute of Eminence scheme was launched by the Government of India to empower higher educational institutions and help them become world-class teaching and research institutions. IIT Madras is proud to have been selected as one of the select Institute of Eminence. A total of 68 research initiatives belonging to 21 identified clusters are presently underway at IIT Madras. As a part of these initiatives, the IRIS webinar series aims to showcase the innovative research being generated to various stakeholders like students, researchers, industrialists, and policymakers. On behalf of the Office of Global Engagement, it is my pleasure to welcome you to the 47th webinar from the IRIS webinar series. My name is Ujwala K, and I am a part of the Office of the Global Engagement. From the Medicine and Biology cluster, the research initiative presenting today is Cancer Genomics and Molecular Therapeutics, led by Professor S. Mahalingam. This research initiative aims at comprehensive genomic sequencing analysis of pancreatic cancers of Indian origin to identify diagnostic markers for early detection and drug targets for novel anti-cancer therapeutics. Our speaker for today is Professor S. Mahalingam. Professor S. Mahalingam is a professor at the Indian Institute of Technology of Madras. He has established a vibrant research group on cancer biology focusing on signaling mechanisms and genomics, and also published a large number of high-impact research articles in peer-reviewed international journals. His group has established India's first national community-based cancer tissue biobank at IIT Madras in collaboration with various medical centers. He is an associate editor of Frontiers in Virology and the editorial board in Scientific Reports. He received an Outstanding Investigator Award for Medical Sciences in 2009 from the Department of Atomic Energy, Government of India. Thank you for being with us today, Professor Mahalingam. We are very excited that you could be a part of this session. Our moderator for today, Dr. S. Parashuraman, is a group leader at the Institute of Biochemistry and Cell Biology. He is also the head microscopy facility, Institute of Biochemistry and Cell Biology, and also the Cell Biology at the National Research Council, Naples, Italy. He is also the coordinator of Italian Advanced Light Microscopy node of Eurobioimaging Network. Dr. S. Parashuraman, we extend you a special welcome, sir. Before we start, a note to the participants, please use the Q&A section to enter your questions and upvote questions that interest you so that moderator can prioritize them later. Over to you, Professor Mahalingam.
Thank you, Ujjwala, for a nice introduction. Good evening to all. And I am Mahalingam. Today we are going to discuss about the research work that is going at the IIT Madras IOE-COE Center for Cancer Genomics and Therapeutics. Movie.
Just share. No, just go for desktop and share.
Sorry for the interruption. So what is cancer? Cancer is a complex disease, and it's usually a combination of abnormalities converting normal cells to become a cancerous cells. For example, if you look at here, the healthy cell proliferation, they have a tightly controlled self-renewal. That means they have a programmed cell death, so they can control the cell proliferation and control the organ size. On the other hand, if you look at the cancerous situation, you will have a damaged DNA. In other words, you may have a change in the genetic material that leads to the abnormal cell division. So eventually you will have uncontrolled cell proliferation. So when you have uncontrolled cell proliferation, these cells are resistant to the control from the normal tissues to maintain the normal tissues. As a result, they divide more rapidly than the progenitor, that's the original cells. And they are less independent of the signal from other cells. So they can grow continuously without a break. At the later stage, these cells can break the normal tissue boundaries. So that leads to metastasis. That means the later stage, they can spread from one site to another site within the body. So these cancer cells, since they have abnormal behavior, because there is a change in the DNA sequence of key genes, which we call it as a cancer genes. That is why all the cancer are genetic disease. And these disease, that means the cancer, is non-communicable, multigenic and multifactorial disease. If you take a closer look at the cell division, and as I said, in the cancer genes, they control this. These cancer genes are broadly classified into tumor suppressor genes and oncogenes. And these two genes, group of genes, they coordinate the cell growth by maintaining their balance in their expression level. That is critical to have a normal cell cycle with the specific control. If there is any problem in the maintenance of their expression level by loss of function of tumor suppressor genes or gain of function of the oncogene, that's the proto-oncogenes, that means they can have a mutation by deletion or substitution of specific amino acid, or they may have a translocation that leads to abnormal cell division. That means there is no control in the cell cycle. That will eventually lead to the tumor formation. So basically, these tumor suppressor genes acting as a brake for the cell division. Just you take an analogy of the car. You have a brake, you have an accelerator. Like that here, you have a tumor suppressor and proto-oncogene. If there is a problem, if the brake is not working in the car, you know what will happen on the road. The same things happen. If there is a problem and the expression or there is a loss of function of the tumor suppressor genes, then the oncogenes, the proto-oncogenes take over. So you will have an uncontrolled cell proliferation. That leads to the tumorigenesis or cancer formation. But if you look at the global situation and Indian situation, what are the cancer are predominant? But if you look at the global situation, to put together both gender, the breast cancer, it constitutes almost 12%. Then lung cancer, colorectal cancer, prostate cancer, and liver cancer, and so on and so forth. This is the incident rate. But if you look at the mortality, the lung cancer comes first. That's the 18%. And interestingly, I want you guys to remember the pancreatic cancer. Here in the incidence figure, you don't even see the pancreatic cancer. It is among the, or it's hidden in the other cancer, 36%. But when you look at the mortality, it is almost like a 5%. It is almost within the top 10 cancer. And if you look at the scenario in India, same like what you see in the global, here also breast cancer at the high incident rate, followed by oral cancer, and cervix, lung, and stomach, and other cancer. But in India, the systematic collection of the data started in 1982. These data are collected by the National Cancer Registry Program. And the ICMR, that is the Indian Council for Medical Research, they estimated around 14 lakh new cases at the end of 2020. And they estimated we are going to have almost like a 21 lakhs at the end of 2015. Now you can imagine how fast this disease is spreading, or the incident rates are going high. Here in this picture, I just put the global population, almost like a 25 to 30 different faces, you can see it, but all are different. On the other hand, if you look at Indian subcontinent, you have so many different ethnic groups. They look different, they behave different, that clearly give an indication that we have a different genetic background. So if you go a little more closer on the tumor, on the cancer, you have a tumor heterogeneity. What do you mean by tumor heterogeneity? So within tumor heterogeneity, you have inter and intra tumor heterogeneity. So for example, here you see 4 patients, they have a similar cancer, but all behaving differently. Though they are similar, same gender, but they behave differently. And if you go a little closer, the tumor within a particular individual or particular patient, and if you look at what kind of cells are there, they're all different, even within the same tumor, same individual. For example, if you look at on this side of the figure, you have 4 or 5 different types of cells within the same tumor. Each cells behave differently. I mean, they look morphologically different, genetic makeup also, there's a subtle difference. And some of the cells from this, they develop resistance for the treatment. So most of the human cancer are encompassed genetically and phenotypically heterogeneous cell population, cancer cell population. That leads to major challenges for the clinical management of cancer patients. So the understanding the mechanism that regulate the tumor heterogeneity is very, very important. So what is required? You need to have genomic profiling technology, that is genome-wide next-generation sequencing, that will help you to uncover or understand different aspects of genomic heterogeneity in multiple cancers of human origin. And this will accelerate the identification of genomic signature towards personalized treatment or precision medicine. So what do you need to do the sequencing? So you need to have cancer tissue samples. Without that, we cannot do it. So a lot of magazine, the newspaper, they publish, you need to have tissue samples for the identification of the biomarker specific to different ethnic population, and to identify or developing the targeted therapy. For that, you need to have tissue samples. Even the Time magazine, they published top 10 ideas challenging the world right now. The biobank, it's one of the top 10 ideas. So keeping that in mind, now also, there's one more thing. You need to have a tissue samples from the patient to understand the genomics. In addition, if you look at the model system that are available to understand the pathophysiology of human disease, we have cell line from human animal origin, but these cell lines are multiplied multiple times and very long time. So there is a change in the genome. So whatever the work you do with that is not going to reflect what is really going on in the human. And in the pyramid next, you have the human cells or tissues in an animal host. So you take the human cells, put it in an animal host, and they try to understand the pathophysiology of a particular disease, in this case, cancer. And the immune system of the animal is totally different than the human. So it's not going to reflect what is really going on. And you have a primary cells, that's the same issue. And you have the humanoid animals. The immune system is different than what you see to the human. So then what is close? So you have a human organ and human tissues. You can directly do all genomics and all kind of work which will reflect what is really going on. And they are closely related to what you see with the human because it directly comes without any manipulation. So keeping that in mind, we have established India's first community-based national cancer tissue biobank with the funding from Department of Science and Technology, Government of India, and IIT Madras, Chennai. This is a class 100,000 tissue storage facility. So here we have a storage facility. Basically, we store all the tissue samples in liquid nitrogen facility and all the nucleic acid at -86. And we have complete pathology facility, tissue pathology facilities. So automatic tissue processors, stainers, and embedder. And we have a sequencing facility. We have NGS, Illumina platform. And to confirm the NGS data, we have a Sanger sequencing facility. And for validating and developing the biomarker for cancer, we have a nanosting facility. So what are the objectives of this new center, the Center for Cancer Genomics and Molecular Therapeutics? Here, we want to do the integrated and comprehensive genomic analysis of the pancreatic cancer from Indian cohorts. We want to develop the mutational landscape and candidate genomic events that are important for pancreatic carcinogenesis, and to define the differential gene expression and transcriptional network in pancreatic cancer, and comprehensive genomic analysis of pediatric leukemia, and development and characterization of pancreatic cancer preclinical organoid model for drug screening towards personalized medicine, and identify specific biomarkers for early detection and target to design novel anti-cancer therapeutics. So using all these genomic data as well as patient information, we'll want to develop the Indian cohort-specific cancer genome database. So we are starting with pancreatic and pediatric leukemia. Why start with pancreatic first? So you know the importance of pancreas. This is a vital organ, regulate blood sugar level by synthesizing two different important hormones, that is glucagon and insulin. The pancreatic cancer is often diagnosed at the very late stage because of its location. It makes it difficult to diagnose it at the early stage. And if you remember in the previous slide, the incidence rate is not as high like other tumors, but the mortality rate is very, very high when compared with all the tumors. So this is the 8th most common cancer death in the world because of poor prognosis. And the median survival for all patients is 4 to 6 months. And the survival rate is one of the worst when compared with all the other cancer. And interestingly, if you look at the mortality to incident ratio, it is 98%. And if you look at in this table, you can see, I mean, I compared 1990 to 2017. The incident rate here in India, particularly in India, 6,800, and the mortality is almost 6,800. And the same thing goes with 2017, 30,000, and you have 29,000 something. That is true for all the countries. That is true for globally. And also it comes to, it comes under the GI, that's the gastrointestinal system. In the gastrointestinal cancer, the incidence of this cancer is very high in India. That is the reason we have selected pancreatic cancer to start with. And so far we have collected almost like 5,000 samples. And this is the workflow. So we get institutional ethical clearance before we collect the samples. And we get the informed consent from the patient. And we developed a specific protocol for collecting and storing the samples, tissue samples. We are doing the sequencing, both tumor and matched normal samples, both exome and transcriptome sequence, because each one will give different information. And we also planning to do the whole genome sequencing for some of the samples. So we'll get SNPs, germline, somatic, driver mutation, inters, and structural variants from exome. And transcriptome, we get differential expression, gene fusion, alternate splicing, RNA editing. All these information will integrate with the patient information, which is critical for development of the database. And all these mutations or changes or alterations will be confirmed using Sanger sequencing and real-time quantitative PCR, and compare with the pathology information and the responses to the drug treatment. That will eventually help us to identify biomarkers. And if you look at the pancreatic cancer stages, so you have a normal, stage 1, 2, 3, and you have infiltration, dissemination, metastasis. You have not even known any marker for this. Even for this, the beginning, we have only few markers. That is also identified from the Western population that cannot be directly applied to Indian population. And you all know why. Because of inter and intra genomic variability or heterogeneity. And for pediatric leukemia, we have collected longitudinal samples. So that means we have selected 75 patients. So far, we have recruited 57 patients. We collect the samples before the treatment and after the treatment, and we follow up the patient. So far, we have followed up almost 3 years. So we have to do 2 more years. And we collect the blood, we isolate plasma and PBMC. This PBMC will be used for whole genome, exome, and transcriptome sequencing by NGS. And why do we want to develop the organoid models or organoids for pancreatic cancer? So this will be very helpful for drug screening towards personalized. Of course, the collection process for developing organoid is different than just the collection process for doing the genomics works. And this is a specific protocol. We collect the tissue, we develop the organoid, this is the protocol. We'll do sequencing of these organoids. Basically, you are not manipulating anything. You just grow the cells in your mouse. And we also do the sequencing of the primary tumor. We compare the sequencing of the organoids and the primary tumor. And based on the sequencing information, we screen whatever the drug is available for pancreatic cancer. We test in vitro with organoids model. Based on the responses, we can give whatever the drug is giving better response that can be recommended for the treatment. This is the work we are in the process. We already developed the protocol. We are working on this. So once we have all the genomic information, like whole genome sequencing, exome sequencing, and transcriptome sequencing, we can able to identify point mutation, indels, copy number variation, structural variation, differential expression, gene fusion, alternate splicing, everything. And based on these information, we will select the samples for epigenomics, like a bisulfite sequencing or chip sequencing. We'll integrate all these information with the patient information, try to identify the functional consequences, and try to understand the cancer pathogenesis and clinical applications. And if you look at the social impact, the deliverables of this, you'll be able to identify a novel pediatric cancer and pancreatic cancer and pediatric leukemia specific biomarkers for early diagnostics, and that targets for novel anti-cancer therapeutics. The pediatric cancer organoids will help to screen the drugs towards personalized treatment. Now you know what kind of modalities they are using, because you have a lot of off-target effect, that's the side effect, because the drug is developed for some ethnic groups and we are giving it here. That is why you see an off-target effect. The success rate is not as high. This will help us. And the development of cancer genome database specific to Indian cohorts. We'll also give different level of training for academics and industrial personnel. And we'll have summer and winter schools and workshop on biobanking and NGS data analysis. And the funding from Minister of Education, Government of India, IIT Madras, Chennai, for doing all this genomics work. And thanks to the Office of Global Engagement, IIT Madras, for arranging this web series. And this is the contact details. And we have project intern and assistant and PDF position. If anybody interested, please, you can mail us. And thank you. If there is any question, I'll be happy to answer.
Thank you, Dr. Mahalingam. It was very interesting. And, I see there is a lot of work ahead in the future.
Yes.
Can you hear me?
Yeah, I can be able to hear you.
Okay. And we have a lot of questions from the students, I suppose. I would like to start with something related to pancreatic cancer. Sure. There is Ayushi Dote. She's asking, how successful is precision medicine against pancreatic cancer, since mortality rate is very high? What about in Western world? Have they tried pancreatic precision medicine? Does it work?
No, it is at infant stage. So because the success rate is low, because we don't have any markers for detecting at very early stage. And resection also, it's very difficult because of its location. That is why the diagnosis is very difficult. They do it at very late stage. That is why the mortality rate is very high. The precision medicine or the personalized treatment, it's the only option. That is what now the whole world is looking for. For that, we have to sequence an individual cancer genome. Then only you can able to identify what is the changes is going on with a particular individual. Accordingly, you have to design the treatment strategy.
Okay, thank you. So there are people who also have doubts about organoids. There are students who don't know what is an organoid and what is the enzyme that is used to digest the cell tissue to get the cells. So can you give a brief telling about organoids, how they are made, so that the students can know?
You have to, like, for example, if you want to develop the organoids, the collection itself is like, you have to collect like the organ for transplantation, because you want to have the live tissue, live cells have to be there. You have to collect in a special growth media, not in liquid nitrogen. Once you have the tissue samples, then you mince it. That's what I showed in the so here. You mince it. The enzymatic digestion, we are using collagen. So basically, you want to have individual cells, then centrifuge it and put it in cell matrix and with some growth factors. The same growth factor cannot be used for all the organoids, all the organs. You have to standardize. You have to give trial and error with the different factors for different organs. Then you wait for, so you will have a culture, cell culture. You are not manipulating anything. That will give you the genomics. I mean, whatever the genomics so far, the people have done for different like colon and gastrointestinal tumors, some of the tumors, the genomics is almost same. Whatever the changes you see to the primary tumor, you see the same. You don't see any additional in organoids. That is why people are trying to develop organoids which can able to like reproduce, or you may be able to get the responses when you do the drug treatment in vitro, what is going on in vivo.
I think this is, you have already answered this question, but still, since there are ethnic differences, genomic differences between, say, Caucasian and Indian, is it necessary to develop organoids based on ethnicity, or there is a universal organoid model? But
when you say
I think this you have answered already.
Treatment, you have to do for individual person.
Okay.
Because the genetics is completely different. Right? You
Okay, and another thing,
inter tumor heterogeneity. So when you have like that, you cannot develop for one person and it may work. Sometimes yes, but most of the time no.
And this is another interesting. Is there any particular reason for increase in cases of pancreatic cancer in recent years? Is it more incidence or more diagnosis now?
No, the incident rate is high. The incident rate is high. Still, the early diagnostic is not really happening for this, because you don't have really good marker for pancreatic cancer detection, because we have only few markers that's also developed for Western population, for a particular ethnic groups, not for everybody.
I think the question is about the increase in rate, because you say in 1999 there were 6,000 patients and now there are 30,000. So is it going up or it's more getting diagnosed better?
No, the level is not like the 6,000, 30,000, 36,000 is 1990. 30,000 is 2017, almost 27 years difference. So the rate is not really increasing very high, but the rate is going at a steady level, steady state level.
Okay. Okay, so these are the things I could find specifically related to the talk, but there are a lot of general questions.
Sure.
One is about breast cancer. What is the risk factor assessment tool most reliable in assessing breast cancer risk in Indian population?
Breast cancer risk
tool, a tool to assess the risk. Like if you are if you are BRCA1 mutated, there is more likely to get something, no? Or if you have a
see, we have some unpublished data, particularly on breast cancer, since you touched about BRCA1. See, I mean, whenever a patient with suspecting the breast cancer, they try to do or undergo this BRCA test, BRCA mutation test. But the BRCA mutation, BRCA1 and 2 mutation test is developed against, again, the Western population. And if you look at the mutation rate in Western population, it varies anywhere from 9% to 21%. Depends upon population you are talking about. But we did the breast cancer sequencing of 200 people. Of course, the number is too less. And when we look for the BRCA mutation, both 1 and 2, out of 200, we hardly see 1 or 2 patients, they have the BRCA1 and 2 mutation. That clearly indicates, but it's too early to tell. As I said, the number is too less. That is not that may not be the right test for our own population, because of the genetic background is totally different. So we have to do sequencing. We have to find out what is really going on with our population, what are the alterations are there, and what is significant. Accordingly, we have to develop the diagnostic tool our own population.
Okay. So right now for Indian population, it's missing, as you say, which is
nice. So,
given that a lot of people-
there are some sporadic, the genomic data is available. For example, like oral cancer, there are 100 patients they have sequenced one group in Kalyani, the DBT Institute, they sequenced and they published. Other than that, I don't see so far any big data is coming out so far. So we have to sequence, we have to find out what is really going on.
Yes, given the high incidence of breast cancer in Indian population that you showed, I think it is essential now that we need to develop our own tools.
Yes, we are also doing. We just we completed 200. Now we want to sequence up to 1,000. So we already collected the samples. We are in the process of doing that.
Another question: how far is stem cells helping in treatment of cancer? And there is also another related question, which is if we provide bone marrow from to a person in blood cancer, can it cure? Which is similar.
We are trying,
can stem cell therapy be
trying for leukemia, not for solid tumors. Yeah, we are trying for leukemia. It works at a certain level, but solid tumors, I don't see things.
Okay. And then is diagnosis with the help of biomarkers more expensive than a normal CT scan?
I don't think so. It will be more expensive. Like, depends upon what kind of markers you develop. You can use a real-time PCR based or nanosting based or sequencing based. I don't think it's going to be that expensive like CT scan or PET scan. I don't think so.
Can we edit the DNA which in the late stages of cancer to bring it back to normality?
No, late stage, you yourself said it's a late stage, right?
Yeah.
So it's metastasis. Metastasis, when you're talking about late stage, is a metastasis, right? Metastasis is already spread from the primary site to different sites. So that means, and in vivo, you know the problem of genome editing by CRISPR. See, they can do with all the prokaryotes, it works really well. In the eukaryotic system, also human system, cell line, it works well. In vivo, there's a lot of ethical issues. And in vivo, like how many cells you will do? Of course, you can do lentiviral based, but whether you can able to do it in correct in all the defected cells, that's a big question mark.
Okay, there is one more on your methods.
Early detection is always better to have early intervention. Yeah.
Okay. Do you have matched RNA-seq clinical and exome sequencing data for all 5,000 patients?
Which 5,000 patients you are talking about?
The close to 5,000 samples that you have collected, no?
No, we have 5,000. We have collected close to 5,000 samples of different cancer.
So for all of them, do you have matched RNA-seq clinical data as well as exome sequencing data?
We have not done all the sequencing yet. We started with the breast cancer for a different project. Now we are doing with the pancreatic cancer. We have not done with other cancer, but we have the tissue samples. We have, like when you talk about 5,000 samples means 5,000 patients. You have 10,000 samples because you need to have matched normal for the comparison. Otherwise, it's very difficult for you to know whether the mutation or whatever the aberration you see, it's specific or not.
So you have collected them, but not done the sequencing in it.
Yeah,
right?
It's a new initiative. We started very recently. Yeah, and we
And then there is the same person also asks another question. PBMCs are a subset of immune cells.
Yeah.
So why are not including other cell types like eosinophils and neutrophils? I don't know whether it is required for cancer type. I have not known about a eosinophilic or neutrophilic based cancer. Do you know?
No, I don't think so. Because you know that ALL, that is a majority, you see it, and you have AML and CML, that is all on PBMCs, white blood cell. That is why we talk about that.
Okay. How is the tumor heterogeneity within a tumor tissue quantified or measured?
See, there are multiple ways people are doing. You can do like immunohistochemistry, like tissue microarray, you can use it. And you can do the tissue micro dissection and you can do the sequencing. And identify, you can do the tissue microarray and identify the different type of cells. Then you can do the dissection, you can take out the cells, you can extract your RNA or DNA, then you can do sequencing.
Have you also, or I don't know whether you have also plans to go for single cell sequencing to see heterogeneity too?
Yeah, we are planning for the single cell RNA-seq. Yeah.
Okay. Yeah. Any study for alveolar rhabdomyosarcoma? Are you all planning to include that in the? I don't know what is the incidence in India.
Rhabdomyosarcoma is not that high incidence. We have collected only few samples. The sample collection, so far we are collecting, like included hospitals in and around Chennai and Pondicherry and some hospital in Mizoram. Maybe if we can expand to other or different parts of India, we may get more samples, but so far the incident rate is not as high like other tumors. But yes, we do. We have collected some samples.
How do we diagnose cancer, any type, at a very early stage?
So, can you say it again?
How do you diagnose cancers at a very early stage? Any type, it's not.
See, very early stage. See, some people, they may not even know that they have a tumor. They have a cancer. They feel different. They have a lot of behavioral changes and pain. So you feel different than the normal behavior. Then try to do all kind of imaging technique. That's what initially they used. Try to identify where it is and what is the stage. But in India, few centers, they are trying to do the molecular pathogenesis, molecular diagnostics. But again, that is all depends on whatever the marker identified for Western population. Again, I'm telling that we need to have our own genomic information to have better diagnostic marker.
I think there are several questions on the same thing about somatic mutation detection in breast cancer. You have already answered them. Yeah. Like in India, it seems to be different and we need to develop. We don't yet have it. Yeah. Am I right?
Yes. See, that's what I gave you an example on the BRCA1. So, BRCA1 and BRCA2. So, the BRCA1, BRCA2, because the mutation rate is high in Western population, they developed. But as I said again, the number, whatever we sequence so far is very less. We cannot, I cannot make an authentic statement
Mhm.
But whatever we did the sequencing, we see even less than 2%. That means I don't know whether the test can be used as a gold standard test for a breast cancer. Based on that, whether you can do the treatment.
Yeah, diagnosis. And this is an interesting question, but this has been there for a long time. I think though the question is wrong, I heard that big animals have less chance of developing cancer. I don't think they have less chance. They have less than expected chance, let us say. Is it true and why?
Can you say it again? Sorry.
Big animals. No, this has been a question for a long time. Like an elephant has much more cell divisions than me. So why doesn't it have a more chance of developing a cancer? No, it should have a very high chance of developing cancer, but it doesn't, as expected. Why? There was a.
That is not true because the size is bigger. You'll get no. They have there is a the pathway is called hippo pathway.
Mm-hmm.
Each and every individual, they have a specific regulation of the hippo pathway. Hippo pathway is nothing but controlling the size of an organ. That depends upon the animal. Does it mean that the bigger animal develop cancer faster than the smaller animal? No. The pathway, it works accordingly. If there is alteration or if there is a deregulation in that pathway, there is a possibility. But I don't think you can extrapolate with the size and the incident rate.
There are also some studies, if I'm right, which found that I think it's elephants, which have more copy numbers of TP53 or something like that. They have
P53, I don't know, because there is a lot of controversial thing is going on. Like, P53, everybody knows it's a guardian of the genome and it's a tumor suppressor. And if it is a tumor suppressor, I'll just throw an open question. If it is a tumor suppressor in some type of breast cancer, and you have a different molecular subtype of breast cancer, you have a very high level of P53 expression, but the tumor is very aggressive, not responding to any of the treatment. Why?
I don't know. It's interesting though.
Because it's a context specific. It works differently. The stoichiometry of the protein concentration is very, very important. Like a couple of years back, we published a paper on Journal of Biological Chemistry. We have proven the mechanism using a breast cancer as a model system. So P53, when the stoichiometry changes, it works in opposite way, like a coin has a two sides. It works in opposite way, acting as an oncogene by activating P21. And the P21, of course, everybody believes that it is it's controlling the cell proliferation, but in certain situation, it alters your CDK's level. When it alters the CDK's level, you will have uncontrolled cell proliferation. So what is the culprit? Culprit is the P53. That is the one is altering the level of expression. But of course, P21 expression is dependent and independent of P53. It is in a context-dependent manner. So the bottom line is each and every, though we call it as a tumor suppressor, yes, it's a context-dependent. Not always, not in all the situation, it's a tumor suppressor.
That's interesting to know.
Yeah.
And then there is one person who's asking.
That is the truth for RAS. For example, in RAS you have, right? Everybody call it as RAS is an oncogene. No, mutant RAS is an oncogene, not the wild type RAS. Wild type RAS is induced senescence and differentiation and cell cycle arrest.
Yeah. There is an oncogene-induced senescence.
It behaves differently in a context-dependent manner. What is the upstream regulator? What is the downstream targets? Accordingly, it works.
Okay. So moving ahead, the one person who is asking for the organoid generation, do we have everything or we do it with the collaboration with Western countries and firms?
No, this is video. How do we do this with the Western countries? Because you have to do the organoids test like as soon as you collect the cells, tissue.
Mm.
Yeah. You do everything in IIT Madras. Of course, some other lab also in India is doing, not just yet the only one is doing, other people also do in India. Like IAC, they do the breast cancer organoids, and some of them they are doing, I mean, we are doing pancreatic, and we are trying to develop colon also. But
is there a way to target cancer stem cells?
sorry, is the way to target?
Cancer stem cells.
See, cancer stem cells is another big area. And they have, I mean, you have a theory on both positive note and the opposite view. And cancer stem cells, the people are trying to use the blood samples from an affected individual, try to isolate and see. And if you look at the stem cell, cancer stem cell population, it's very, very less in the blood. And you need to have blood samples from different time point. And some of the publications suggest that if you want to have more cancer-related stem cells, you need to collect the blood 5 to 14 days after the surgery to have more number of cells in the bloods. But each people are using different protocols and try to isolate, try to sequence what is really going on, and also try to understand or try to correlate the number of cancer stem cells with aggressiveness or responses. People are working on that. This is infant stage.
Coming back to pancreatic cancer, given that it's very hard to detect because it's usually detected at the very late stage, no? That's what you told. Are there any early side effects that can point that a person might have pancreatic cancer and he has to go and test?
See, I mean, you'll have like, like a nausea, you have stomach pain, you feel, less appetite and lethargic. That is what so far the people are talking about it. And since your insulin level, you'll have a fluctuation in the insulin level. That will give you an indication.
Are there any plant-based medicine available for treatment?
Plant-based
alternative complementary treatments.
I hear a lot that homeopathic Siddha, but I mean, to be very honest, I really don't know whether that's really, I mean, of course, there may be an effect, good effect, but then one have to do the systematic analysis to identify what is the active ingredient and try to develop. Yes, it might work better, but one have to work on that. But I hear the news from the people, yes, be treated with homeopathy and be treated with all this and we have a cure, but I don't know how far we can go with that, just with the plain crude plant extract.
Is there any Ayush program on this to identify?
Yeah, that program is going on in India. Yeah. The Ministry of Ayush and the Ministry of Health, they initiated a lot of programs and developing this and rejuvenate the whole thing and try to identify active compounds from different plant origin and try to screen what may be the potential active antiviral compounds. Those kind of stuff is going on. Yeah, I've initiated.
Okay. And then another question, when you go for personalized medicine, you sequence the DNA and everything, do you take into account all the polymorphism present in the person or you focus on oncogenes or something specific?
Oh, you have to take into account of everything. And you have to look at the pathways, you have to look at you have to correlate everything. And simply not only looking at only oncology. You have to look at the consideration of everything. And compare and significance, everything you have to look at it.
How expensive do you think this personalized treatment would be? Like you get the patient, you sequence, you find the genes, you do organoids, you find the appropriate drug. How much it will take? How expensive?
How much it is? It will be expensive, but I really don't know how much it's expensive. I don't have any answer for that figure. How much? What is the cost for that? It's everything in infant stage, so it will be definitely. It's pretty expensive. It's not. But when you go for a personalized medicine, it's a tailor-made treatment for a particular individual, right? So you sequence a particular individual and you look at what exactly the changes is happening at the genome level. Then you test it with the organoids and what drug is working. Suppose if none available in the market is working, then what is the pathway is deregulated, whether any other chemicals or any other drugs can alter or affect that particular pathway, then we can test it in the organoid model. So you can avoid all kind of side effect before we go to the patient treatment. But of course, we have to do it as quick as possible. We cannot wait forever.
Another one is, since you are talking about pancreatic cancer, is having diabetes, does it make it having more risk to develop pancreatic cancer? Is there any association between diabetes and pancreatic cancer?
I don't think so, because there are so many diabetic patients than cancer patients. If there's a high chance for diabetic people develop a cancer, then the situation is supposed to be worse, which you don't see it here. I don't see there is an association, direct correlation.
Okay. How much time do we have more? Do we have enough time, first of all?
So we have time, so you can please go ahead with the questions.
Okay. There is one question on which is better, using organoids or using microchips? I don't know what she means by microchips, but I know of organ on chip.
I think probably they are talking about tissue microarray, or they're talking about microarray. Microarray is totally different.
Yeah.
Tissue microarray is totally different. Organoid is totally different. Organoid is basically you are growing the tissue in vitro without any manipulation and try to understand the pathology and use that for screening the drugs towards a personalized. Whereas microarray use RNA to understand or to identify differential expression of particular genes. That's it. And from there, you have to use the tissue samples to check whether that is really happening or not. If it is, how much, whether it is significant or not. You cannot really equate microarray with organoids.
Yeah. And then another interesting one. Can a single tissue sample reveal heterogeneity, complete heterogeneity in the patient, cancer heterogeneity? Yeah,
see, for example, if you have a pancreas or you have liver and you take liver section and you see whether there's a heterogeneity or not. Of course, it's a single organ for a particular individual, right? It can.
I think the biopsies are usually small parts of the big cancer, no?
You can take a biopsy and you can look at it. You can take a multiple biopsy in different region of the same organ and you can do the tissue microarray and you can check it. Yes, it is possible.
And then there are several questions, meaning not one, about what is the state of the art of using CRISPR technology in cancer treatment. I think you already answered that. It's very difficult, but
it's difficult.
But I have not seen much on
in vitro. People are trying to develop, but my personal opinion is it's very difficult to use it in vivo situation, because when you said it's advanced stage, it is spread in a different site or different organ in the body. So targeting, it is an issue.
Another one, I don't see many questions on it. What about CAR T cells? Now that you're sequencing, biomarkers present on the personalized, biomarker present on the particular patient, particular cancer from particular patient. Is it possible to develop efficient CAR T based therapy tailored to this patient?
Yes, it is. Maybe I mean, CAR T cells now it is becoming popular, and that most of the time they try to use it. The CAR T cells, they try to use it for like leukemia and some solid tumors. Yes, but how efficient it will be for solid tumors, I think we need more work on that.
How transmission? I think this is, we can leave it. What does it mean that PDAC mutations are not found in Indian population? It's from your slide.
PDAC mutation. Oh yeah. See, some of the mutation they found in Western population, then they try to do targeted sequencing. Whether the particular mutation you see it with the, not like a whole genome sequencing or whole exome or other sequencing, just targeted sequencing. Like as I said, for the BRCA mutation in the breast cancer, like that here they don't. But also give you an idea that, give you an indication that the genetic heterogeneity. So that means you got to know what is really going on for your own population.
Yeah, again, I think you just now answered about CAR T. There is a question about effective immunotherapy now that we identify potential biomarker.
Effective immunotherapy, it works very well for for a breast cancer. It works really well, like Herceptin. It works very well. So for that, we have to identify a specific target. So when you identify, yes, based on what you see with breast cancer, yes, if you can identify a specific target for a particular population, it will work much more potently than others. Yes.
Okay, another question. The drugs that you identify using organoid models in vitro, what is the chance or probability that will also work in vivo?
You have a high chance because you took this tissue from the same individual, not from different individual. You took the tissue.
No, I think what we are talking about is toxicity, because in an organism there are also other cells and tissues, which will also be affected by this.
That is the problem with the cancer treatment. Whatever the chemo you are giving, you all have a side effect. Not just only the cancer cells are getting affected, the normal cells are also getting affected. That is why you have a hair loss, you have some appetite, weight loss, all kind of stuff. Sometimes you get diabetes, all kind of, because of side effect. That's not specific. Whereas the immunotherapy can be specific to a particular target. If it is overexpressed in a particular tumor and you can use an antibody against that, then the treatment is specific. That's called targeted therapy.
Okay, based on whatever you have proposed here, cancer genomics, etc.
Yeah.
Do you see that there we can evolve biosensors? We can develop biosensors for pancreatic cancers.
Biosensors
in the future.
Good question. Maybe I cannot give direct answer whether it is possible or not. Maybe yes. Biosensor based, yes, you can. If you can able to identify a specific biomarker for a specific population, yes, you can.
Okay.
If you can develop that, then it can be high throughput screening. Yeah, we can develop a high throughput screening. Yeah.
It will be interesting to develop something very simple.
Few labs they are working on.
Okay.
I don't know anybody working for pancreatic cancer, but I know some lab they are working in US, some lab they are working on colon and lung cancer. Yeah.
Among the types of cancers that you have discussed here now, which one is which has more probability of getting transmitted in a family? I know breast cancer does a lot, but I don't know about others.
But some of the hereditary mutation, you see it with breast cancer and ovarian. And some situation, you see it like stomach cancer and oral cancer.
They're inherited, meaning the mutations go.
You see it's like they see it in the same family. All the generation they get one or the other tumor. Some situation the same tumor. So, but there is no systematic study. The same mutation is present. That is why are because of behavioral changes or environment where they are, or some infection due to the virus infection. Okay. But there are hereditary mutations they identified. And they proposed, of course, the well-known example, you have a breast cancer, you have ovarian. Now there are few data publications are available for stomach cancer and oral. Whether it is true for all the cancer, we need more data.
Can you go ahead? This I don't understand the question either, but the person asks, can genetic biomarkers overcome individual-based epigenetic regulation?
Genetic biomarker, no, that is totally different, two different questions. Epigenetic modification is different than the genomic-based biomarker, what you're talking about. So here, using exome and transcriptome and whole genome, we try to identify what are the modifications happening at the aberrations happening at the genomic sequences or the expression level. But epigenetic modification, yes, that can regulate the expression status of the gene. But that modification is totally different. You cannot really correlate with that.
This is also an interesting thing. Right now, the treatments in India is based on what they found in the Western world. Cancer is simple. So the question is, does it worsen the condition of Indian patients since the treatment is based on whatever it has been done on Western population? If we use the same treatment, will it worsen the condition rather than making it better? Is there any?
It works efficiently for some individual, but it's not working for some individual. I don't think you can call it as a worse. Based on the condition, based on the situation, whatever the treatment is available, that's what is going on. We cannot say it's worse than the situation. No. I mean, wherever it works, the survival rate is very good, right? For example, breast cancer. Early detection and some, depends upon the stage of the cancer. They go for surgery, or if it is, for example, in pancreatic cancer, if this tumor is too big, not able to do the surgery, they go for neoadjuvant or adjuvant, or after the surgery, again, they go for different treatment.
Okay. The person is asking about the best diagnostic method for cervical cancer.
Best diagnosis, they do the pap smear test, That is the screening is going on. I mean, that is the non-invasive and much faster. They can able to see, is there any alteration?
This is, I think you made it clear, but still the person has doubt. Sir, you collected normal sample from patients or healthy people coming to the hospital?
No, we collect from the patient.
Normal sample to non-cancerous part, no?
No, from the same affected individual, we collect the tumor tissue and the normal tissue, pathologically normal tissue. Because when they do the surgery, they do the marking, right? When they do the marking, it's very difficult to exactly do that because they don't want to leave any tumor tissue. They take some normal tissue also. So we collect the normal tissue also. Then only we can able to compare. Of course, that may not be now. People are thinking even that is not a normal tissue because you take the tissue from one organ, right? So you may see some alteration there also. So when you compare it, you may miss out something. So there now people are thinking we have to take a normal tissue or compare the genome sequence from the tumor and the match normal from a particular individual to the non-diseased tissue or the tissue from cancer-free patient. But there may be some differences. So it's a big thing people are trying to work out.
It's a more, what I'm going to ask is a more technical question. The person asks, what are the good preserving conditions for a fresh excised breast tumor, which doesn't affect DNA, RNA, whatever protein quality? What are the conditions you use to preserve them when you cut out the tumor?
So we developed a specific protocol to preserve DNA, RNA, and protein because we do whole genome, exome, transcriptome, and proteome. So we collect the tissue samples in liquid nitrogen, and the time in between the tissue goes to between the surgery and put it in the liquid nitrogen, that is very, very critical. So if you can able to put the tissue within 10 minutes after the surgery, that would be the best. That means you have to coordinate properly with the surgeon to get the samples to put it in the liquid nitrogen. So that way you can able to preserve everything. That all I think is fine. But for RNA, some people directly collect the tissue in RNA later, but that cannot be used for DNA as well as for protein. But if you do it in liquid nitrogen, yes, you can do for that you can do for everything. Even it's good for metabolomics.
Now that you have told there are genetic differences, no, in Caucasian versus Indian population, will the generic medicines that are used will still work in Indian population?
Sorry?
There are genetic differences between, say, the Western population and Indian population, no? Yeah. Genetic differences, polymorphisms, whatever. So the generic medicines that work in one population, Western population, will they also work here? Let us talk about cancer in this case. There are generic medicines like 5-fluorouracil, no, which is usually used for. No, I'm just giving, this is not the question, but I'm just giving. Since we are talking about cancer here, do chemotherapeutic agents that are used in Western thing, will they also work in Indian population?
This is the big question we asked from the beginning. That is why we are trying to do this kind of work, you know. So the Western population, the success rate is very high because they develop against a particular target. Whereas here you don't know whether the same target is altered or that is a deregulation of the same target or different target. And we all know that tumor is a multigenic. It's not a single gene is involved. It's a multiple gene, multiple pathways. So what is the pathway is deregulated? Maybe if it is the same pathway is deregulated for some individual, it may work. You cannot make a general statement that yes, whatever it works there, it will work here the same manner or same efficiency.
And then there is one more interesting thing. What about nanobots? Because the questioner asks, since even if you remove cancer, there will be some cells left. Yeah. What about using nanobots to treat them?
Nanobots?
Like small robo. I think the nanobot we use is CAR T cell, no? CAR T cell is a nanobot, and technically. Yeah. It's not a machine, but.
See, the residual tumor is another big area, gray area, people are started working on that. Whether there is a residual tumor, if it is, how to treat. But the nanoboards, I really don't know how efficient it is for blocking the residual tumor cells after the surgery. Now, people now, they are trying to develop, they are working, many labs, they are working on the method to detect the residual tumors. If we develop that method, then it is possible, right? How much it is there, where it is there, what is the stage, and everything. But again, that is depends upon the stage of the tumor, what you're talking about. If you're talking about advanced stage, if it is a metastasis, and if you do the surgery in one or whatever the primary organ, it's already the lymph node and other organs, then how is it going to work? It's an issue. So depends upon the stage. And we have to develop a specific method for detecting the minimal, they call it as a minimal residual tumor. Yeah.
I think this is more a general thing. Several people have asked, since we are dealing about pancreatic cancer, what about insulin levels in these patients? I know they are altered in these patients.
It is altered. It is altered heavily. Yeah.
Now that we have talked about CAR-T therapy, CRISPR and everything, someone is asking about gene therapy. Is there gene therapy available for cancers?
No, I don't. Not that I know of that. Because the gene therapy, you have to use a virus. No.
Yeah.
And virus,
maybe if,
like suppose if you're using adenovirus, then you have immunogenicity problem, because most of the viral protein they create a immune activation in the patient. So that is why most of the gene therapy work is stopped with using adenoviruses. Yeah, adeno-associated viruses you can use, but efficiency of targeting is a big question mark. And retroviral, lentiviral based people are working on that, but isolated organ it is. Yeah, organoid it is working. Individual, the cells, primary cells in vitro it is working very efficiently, but in the animal model also it is working. But treating cancer, yes, still we have to do a lot. Hoping for that.
When you do organoids, do you also create a microenvironment that is similar to what is present in the patients?
Of course, that is what we use as all kind of matrix, everything. Then only it will grow, otherwise it won't.
Nanobots. How far is personalized treatment in India? How far is it developed? Is it used, or it's in a very...
Even in the Western world, just started. I don't think India is anybody's doing it now. Because first we have to know, we have to develop the complete pipeline and to prove that it is working, right? So for that, we have to develop everything. So it's an infant stage.
There is a person asking about what is proto-oncology. I've never heard about it. Do you know about it? Proto-oncology.
Maybe it's proto-oncogene, probably. I think maybe proto-oncology, not proto-oncology.
Okay, it could be.
Yeah.
I think proto-oncology you can go and read. So let us go ahead. What is the longevity of these organoids? How often they need to be passaged?
No, you can preserve it. No, you can put it in liquid nitrogen. You can pass it and put it in liquid. If you do it very frequently and for a long time, then you will see changes. You don't want to have changes. You want to basically reproduce what is happening in vivo, right? So you don't want to grow for longer time and multiple times. You develop maybe 1 or 2 passage, then you freeze it. Then whenever you need to do the screening, you can take it. You cannot continuously grow for a longer time.
There is the age-old question on genes versus environment as a cause of cancer. What do your opinion? They're asking for your opinion on it.
Well known, enough.
Yeah.
That is well known.
Yeah.
There is an association.
Hmm.
That's what you call it. It's a chemical carcinogenesis, right?
No, they wanted your opinion, so I thought I'd put forward. This, I
Yeah.
think you can anyway find it if you go and read. What about nutrition?
See, I do agree. I do agree that the chemical carcinogenesis is one of the major problems. So the environment is playing a major role on that.
Especially for smoke, no, lung cancers and smoke.
Because
Or
not only lung cancer, you get oral also with the smoke. Yeah.
Or oral cancer and tobacco chewing, no, these are well established carcinogens.
Well established, yeah.
And there is one more about nutrition, because nowadays it's coming up, no, starvation, which is low caloric diet, which is known to slow down aging. There have also been studies on their effect on cancer. So what do you think about nutrition and cancer development?
I would look at the other side of the coin of the nutrition, because nowadays many people, they try to use a preserved food, right?
Mm-hmm.
So preservative is, they are using chemical as a preservative, right?
Mm-hmm.
So there is a possibility. That is what is happening with the Western world. That is why the incident rate is very, very high there, because they use preservative. Whereas in India now it started. You look at 10, 15 years back, the incident rate is not as high like what you see today.
There is a question about circulating tumor cells and their utility.
We already discussed that.
Pedigree analysis. Do you know about epigenetic therapy for pancreatic cancer?
Epigenetic therapy? Epigenetic therapy? I never heard of epigenetic therapy. Like, the people are trying to look at epigenetic modification and try to find out what is really happening. But epigenetic therapy, yes, it is a new emerging trend, emerging stuff.
I know there are some drugs which are targeting.
Going to be, how efficient is. See, it's again is boiled down to a particular population, right? What is the modification you see to the particular? It's not going to be the same for everywhere. I can't expect the same treatment is going to be the same manner for every population. Yes, the people are trying to use epigenetic therapy, but for particular on pancreatic cancer, I really don't know whether how efficient it works.
I think we have covered almost most of the questions, which are some of them I've left because they are like, is cancer genetic? But after all these things, this is obvious, it is genetic. So I think we would maybe we can end here. Is that, if it's okay, or is there something else need to be done?
Fine. Anyway, thanks a lot for taking.
Maybe I can. I also have one question, because since I'm also working on this TCGA genomics. What I read, which I'm not I'm more a biologist than any pathologist. So I heard that while collecting samples for pancreatic cancer, which is very special case, like unlike other cancers, the number of cancer cells in the sample is very low.
Because the
so
is very low.
Is it you take care of this in your sample collection?
No, this is very. It varies. It depends upon the size of the tumor. Most of the time, you will see the tumor in the head region of the pancreas, not on the body or the tail. I mean, not means is not like that. You don't get the cancer on the body and tail region, but you do get. But the incident rate is very high in the head region.
Okay.
But the organ size itself is too small, and most of the time they don't go for surgery, they go for biopsy. So we get less, and we go for multiple biopsy samples and the biopsy. So we get, so far enough to do exome and transcriptome sequencing. Of course, we are not able to do proteomics because the sample size is too low. Yeah.
Okay, so thank you very much, Professor Mahalingam. It was very insightful, and I learned a lot. And there are several questions about whether they can have access to your slides. Is there a way to get these slides? People have asked.
I think this will be available in YouTube, and they can watch it.
Okay.
Yeah.
And then there's a lot of people who want to join your lab. So I think they can find your contacts directly, and they can write to you, I suppose.
Last slide, that the contact number and email address is there. So they can.
Okay. So they can contact you.
Yes.
And it's a very important project, and good luck with your studies, and let us keep in touch.
Thank you.
Thank you everybody for and asking the questions.
Thank you very much for particularly Parasuram for taking his time from this quarantine and being a moderator of this. Thank you so much, and thanks to all the participants for taking their time and attending this seminar. And thanks a lot for the Office of the Global Engagement for arranging this talk in a very nice way. Thank you so much.
Okay.
Thank you, Professor Mahalingam. Thank you very much for the talk. Particularly, I think a lot of people were really surprised to know that pancreatic cancer, despite of having such a low incidence rate, it has a high mortality rate with a survival chance of only 2%.
Yes. It's very high because we're not able to detect at the early stage. Yes.
Thank you for your talk, sir. We have had the audience were very engaged with the topic, and the question link, the questions, the variety of questions they've asked. I think it was really an interesting session. Thank you, Dr. Parshuram, for moderating this session wonderfully. We thank all the audience. There are 2 parallel sessions that are going on right now. You must be having access to those on the link tree. So please join us to watch more webinars under this cluster. And we look forward to see you all. Thank you, sir.
Thank you.
Thank you, everyone. Good night.
Thank you for the opportunity.
Thank you.
Bye-bye.
Thank you, Professor.